Hello, all of the scientists of EDEL 433. I hope all your experiments went well. Today I'm here to share what I've been up to the last 4 weeks, and I'm eager to share my results as well. So here is a picture of me holding a glass jar, all happy, on a really hot summer's day when I went out to conduct my science experiment on the water clarity of surrounding bodies of water in St. Albert. I chose to do my experiment on comparing the different water bodies in the city of St. Albert where I live. This occurred one day as I went for a walk, and I noticed that the river, the wetlands, and natural lakes and man made lakes in my area all have unique characteristics, and the water quality was different. So I decided to test the clarity of the water in different locations around the city. And so this is me going out. This is a picture from the river collecting a water sample. So let me tell you all about it. I live in the city of St. Albert and it has many walking trails. And I walk and I ride my bike and I enjoy all of these places. Most of these places I go by have a body of water, and my curiosity started from this. I wanted to see the differences between all of these different areas that I spend so much time in. So, this is actually a map of the different locations in which I took water samples from. For the purpose of this experiment, I took two samples of water from each location to compare them at the end. And the locations consisted of Lacombe Park Lake, which is a man-made storm water lake, from the Sturgeon River, which is a river that runs and cuts my city in half, also from Big Lake, which is a huge natural lake, and the last one was from the wetlands. Now, my hypothesis and focus on the experiment was comparing other bodies of water to specifically a wetland body of water. So, my inquiry question became, how do bodies of water in wetland ecosystems differ in clarity and quality from rivers, lakes, and stormwater bodies in my city? This question was sparked off of a natural curiosity for enjoying this boardwalk system in my local wetlands area at Lois Hole Wetlands Park, and always noticing that the water is super clear. But this actually surprised me because when you think of wetlands, you think of bugs and animals and plants and weeds and maybe algae. And I thought, how can the water be so clean? So I did look into it and I learned lots about natural wetland systems and how they're natural filtration systems that keep their water clean. So I wanted to test if this experiment was true, and I was going to compare how other bodies of water are different, maybe dirtier, and don't have this natural filtration system that keeps the water so clean. Feel free to pause the video here so you can read my formal hypothesis stated in my experiment report. Now, a problem I ran into was defining what clarity means, or what it means for a water sample to be clean, and to categorize differences that I could observe about each water sample. Initially, in my proposed experiment, I didn't have a proper definition of what I was looking for. And in the feedback I got on my experiment, a lot of people or my fellow scientists in the class told me that I needed to specify what it meant for water to be clean. So here was my finalized categorization of what I was looking for with each water sample. And everything from the clarity to particles to chemicals to anything biological and living, the color, odor, and pH were all things that I put into a finalized chart of observations that I had to record of each sample. All right, so this is me going off into the wild and collecting my samples of water. So there were four locations, and I took two samples from different areas of each location. And I also made some initial observations in case those were contributing factors to the end results. And in some cases, they were. All of this will be written out in full in my report. But as an example of an initial observation, the last picture, or the one on the bottom right, was me at the wetlands. And when I had been there before the class started, enjoying the nature there, there was no green stuff sitting on the surface of the water. And when I went to go collect my water sample, I was shocked by all of these little green, like lily pads that were just floating on the surface of the water. And I was like, "Oh no, this is gonna affect my hypothesis," because I thought this water would be the cleanest. Um, and yeah, little things like that did occur at each locations. That was definitely the most significant one. But I had lots of fun trying to crawl around and walk through awkward places to try and find good sample locations. So this is me on a very hot summer's day collecting my water and walking through town with a bunch of jars of water. All right, so here are my 8 jars side by side in my chart A photography observations. And I also had a chart B, which had my written observations based on all of the characteristics of clear water. And I'm going to go through some significant findings about each of the samples. So, for the Lacombe Park lake samples or the man made lake samples, there was a lot of particulate in the sample, which was including like dead leaves and sticks and sand and dirt floating around in the sample. Um, Everything was non-living, but it just had a bunch of small little pieces of dirt in the water, I guess you could say. And then on the surface level, there was some green algae that settled at the top. So like this dirty greenish settlement that just floated on the top layer. It was very thin, but it was present. The Sturgeon River sample was very clear. And at first, I thought this was going to be the cleanest sample. But yeah, both samples very clear, especially in color. A lot of the other samples had like a slight yellowish color to them, but this one was perfectly clear. And there was a lot of small, very small non-living particulate matter in this sample. So it was hard to tell based off of the photo here. But when you look very closely, and I even had to bring in a magnifying glass, there's so many small little black specks. Now they're non-living because they're just floating around, but very tiny, small little speculants in this one. Um, the big lake sample was the hardest to capture, and I talked about how this sample in particular might have been an error on my part, but I couldn't find a great place to take this sample from. So the cleanest place was the shoreline. It was accessible and it was fairly clean, and I had to kind of really put myself in there to get this sample, but it still came out really dirty because there was a lot of sand and dirt just on the shoreline. So I said a more fair sample might have been from taking the sample in the center of the lake, but I had to work with this sample. And so, as you can see, this one just was very dirty, very sandy. Now, there was a lot of uh, plantation on the shoreline here, and I even saw a decomposing muskrat on the side as well, um, which probably affected the sample. And then the last one was the wetland sample. So the water was very clear, but it had a slight yellowish tone, as well as one had a bunch of plantation at the top, and the second one I went to find a more clear spot. One notable thing about the wetlands, though, was that there are living organisms in this sample. When I looked at the sample very closely, there are little tiny bugs, maybe little tiny organisms swimming frantically around the sample. So I was quite fascinated to learn that there were these little guys swimming in my sample, just doing their thing, and they were still alive a week later. So I was very impressed when I went to discard my sample. And none of the other samples contain those little living creatures. So it was very unique to the wetland sample. Now, I made a ton of observations about each sample, and they were recorded in my observational chart. And I will put access to that chart. So if you're watching the video, you can go and read my full report to learn all about the samples. But again, the most significant part was the living things in the wetland sample. Tiny, tiny specks in the Sturgeon River sample. Just the big lake sample just ended up being dirty based on the location I took it at. And then the Lacombe Park sample just had lots and lots of like algae and surface foam and then non-living particulate in it. So a significant find of this experiment was the pH of each water sample. Initially, and if I'm being honest, I thought that it wouldn't tell me much about each sample because I was assuming each sample was going to be a 7, water, neutral. I didn't think otherwise. But to my surprise, I found that most of the water samples are actually pretty acidic, except for the wetland sample, which came out as neutral at 7. And what I observed was the more particulate contamination there was in the sample, the more likely that it was to be more acidic. So, for example, Big Lake, there was lots of dirt, roots, grass. Lacombe Park Lake, there was a lot of very small particulate in that sample and a little bit of green stuff at the top. And then the river as well, that one had a lot of particulate in it, and it was taken near an algae sample. And the wetland sample, the only particulate in that sample was those green leaves, or the green leaves in the first sample, and then the second sample actually had living things in it. And so that actually struck my curiosity further. I was like, oh, wait a minute. There actually might be some form of relationship between the wetland's ability to have these little organisms living within the sample and have a neutral 7 pH. Maybe there is something going on in the wetlands ecosystems where it's consistently trying to maintain a neutral pH so it can balance the life. Because why are all those little green leaves so bright and vibrant and alive floating on the surface, those little lily pads? And why are these small, tiny bugs still able to remain alive after 1 week of the sample sitting? And that was the biggest find I found was the pH of the sample actually corresponded with its ability to maintain life. Keep the leaves, keep the little lily pads alive, and keep the little tiny bugs alive in the wetland sample. Where all of these other samples were affected by some sort of bacteria contaminant that made the sample more acidic based on my observations. So I drew conclusions based on my observation by comparing all of the jars. And here is a relevant comparison taken from my report on the two cleanest or clearest samples. The one on the left is the wetlands, and then the one on the right is from the river. And the wetlands sample is a little more yellow in color. This sample was unique because there were visible living organisms. There was settlement on the bottom, some sort of like particulate that was like sticking to the bottom walls of the jar, but it was quite minimal. And then the pH was neutral. Then the river was clear and there was no color. It was very clear. There was particulate in this one, non living. You can tell it's living because it's swimming frantically, or non living is just floating naturally. And then there was a high quantity of particulate, but it was very small, very, very small, like little itty bitty pieces of dirt or dead leaves or sticks. Seems that the river might have broken it down very, very small, but there is still a lot of it. And then it was more acidic. Now, oh, and also the algae on the top that settled, or like not green foam that settled on the top, and it came out to be more acidic at a pH of 6. Now, I thought this was a neat comparison because just looking at the two samples, if I had solely based my experiment off of observations, then I may have come to a false conclusion because based off of visual observations, the Sturgeon River or the river jar looks cleaner. But it's not cleaner because it has such a high acidity. And that's why pH became a really relevant indicator to tell me a lot more about each sample. Because we may think that rivers are the cleanest because they're naturally flowing and breaking down everything that's following the current, but maybe that contributes to an overall higher acidity of the sample because there's so much more small little broken down pieces. Whereas the wetland sample, again, is trying to maintain life, so it came out to a neutral pH. I just found this comparison very interesting. And then it drew me to the conclusion to which I can't really determine which sample is the cleanest because it may be more relevant to just say, okay, what's the pH of each sample? Because the wetland sample is actually cleaner based off of the pH. So it became a really interesting experiment in that way, but yet still so much to learn from. So, what is going on with our wetland sample? What did I learn from the wetland sample? Well, you can see on the screen, this is what I wrote in my formalized report. And the biggest differentiator factor was its ability to sustain living things, these small little organisms living within my sample, as well as it has a neutral pH. Whether that's a correlation or not does need further exploration into another experiment. Maybe we can take more consistent samples and see if this remains the same. But none of the other samples had living things in them, nor a neutral pH. The other samples just had very high populations of particulate um, dead things floating around in the sample, algae, foam, odors to them. Where this wetland sample, even though it wasn't the clearest visually, it was a neutral pH and had living things, which was just so fascinating. And so um, there must be some sort of biotic and abiotic relationship going on, almost like a self regulating system in this water sample. A mechanism that's always trying to balance itself in a symbiotic relationship so it can continuously sustain life. And that is just an initial observation about a wetland sample. There's so much to be learned about how that occurs and how that works to sustain the sample and the living things in the sample. So, as an extension of the next step to test our explanation further, would be to gather more consistency on whether there is a correlation between a neutral pH and a wetland's water sample's ability to maintain life, whether that is plantation or small little bugs and organisms. So, taking multiple samples and seeing the consistency of this will tell us a lot about how a wetland sample regulates itself and functions. And we can even go further into taking them in different times of the year and so on and see how that relates. Now, to relate this to an elementary classroom, I found a very unique video on YouTube from another teacher who was teaching her students about the filtration systems of a wetland. And this would be where students build their own filtration system based on raw materials gathered from a wetlands area. So, say they collect leaves and grass and sticks and stones and they use that as a filter and they try and clean out dirty water and see if it becomes more clear as they put it through their filter. So these are two kind of extensions we can use to one, test the validity of our own experiment, and two, further extend on learning about the significance of a wetlands ecosystems and its natural filtration and regulation system. Well, thank you everyone for learning with me, and I'm excited to see everyone else's final reports and presentations of their findings. I learned so much about the importance of wetlands ecosystems, and I've also learned that there's so much more to know and study and learn about wetlands. And I definitely fulfilled my curiosity need, and I'm looking forward to a lifelong career of teaching and curious students learning all we can about our beautiful wetlands and home areas. Thank you again.